Hello and welcome to BIOS Frontier Science. I'm Kate Shannon, and today I'm excited to welcome Alex Marson, a professor at the UCSF School of Medicine. And to help host this episode, I'm joined by my colleague, Chris Gaba. Alex, to kick things off, could you please give us a quick introduction to yourself and what you're working on?
Sure. Well, first of all, thanks a lot for having me today. My lab studies the gene programs, the DNA programs that control the functions of cells in the immune system. And we have been using and developing CRISPR-based technologies to go in and rewrite DNA sequences in human immune cells to understand how DNA sequences control the behavior of cells, and then also to think about how we can rewrite DNA sequences in that code of immune cells to transform them into more effective treatments for a range of different human diseases. I was trained as an infectious disease doctor. I don't see patients anymore, but now really think about basic science and then how do we translate that into new immunotherapies for human disease.
Awesome. And before we get too deep into the specific projects that your lab is working on, something we like to ask our guests is, do you have any North Stars that guide your research or tie it all together?
It's a great question. I think I have a few. I think one. I think about a question that comes up a lot is, I'm involved in some biotech. There's a lot of opportunities to do biology in different sectors. Why do things in the academic sector? Why do we stay in academic research? Why do I stay in academic research? And there's a few things that I think are really central to that decision, and that serve as North Stars in thinking about what is the academic mission of my lab and the group and the culture that I'm helping to build. One is to create, a environment that's fundamentally enabling for new discoveries. And so that's broad, but I think that there's a sense that we're in an era where so much is possible. But fundamentally, we're interested in creating a place where people can ask questions where we don't know the answer, and where we can, I think it's something that is fundamentally sets us aside as humans, this ability to ask questions about the environment that we're in and interrogate it. And I don't see any more exciting place than to do that and direct it against understanding the components of our body and what determines health and in a number of different settings. And so, that is really the sort of fundamental principle. Can we engage people's most creative sides to ask fundamental questions about what keeps us healthy and how can we restore health in the setting of disease? So that is one thing, just to fundamentally do everything we can to encourage creativity and play and discovery. And I think that can't be overstated. A second is to think about, can we sort of earn that luxury of exploring space and making new discoveries by coupling into opportunities to improve human health. And so I think that's, where I told you I was trained as a physician scientist. I was trained as an infectious disease doctor. I don't see patients anymore, but that has really left a mark on me and sort of programmed me to look for opportunities where there might be opportunities when we see new discoveries. Okay, can that be harnessed in some way to potentially either understand disease better, and in an ideal world, really treat disease better in a way to improve human health? And then, the third North Star is really around training people, creating, and that's in every sense. I think that 1 and 2 are fundamentally enabled by the people that we weave together, and creating collaborative opportunities. And then, I think the joy of being in an academic setting is getting to work with people at every stage of their training, people who are still in high school, people coming right out of college, to career scientists. And, I continue to learn from people who bring new perspectives in each one of those. But I think that the lab environment when it's running smoothly, creates a learning environment where people are training each other in all sorts of directions. And so I hope that people leave the lab sort of feeling that they've taken something about that experience, and that carries them forward, and then that continues to ripple forward in how they train people in their labs or in their future jobs. So I don't know. That's 3 North Stars. I don't know if you're allowed to have 3 North Stars, but I guess somehow that's sort of some sort of celestial navigation that I try to use to guide, to think about what the environment we want to create in the lab.
Well, I think you're more than welcome to have 3 North Stars, particularly if they're as good as those. I'm really excited to dive into this and hear more about what you're working on. So to kick things off, I'm going to pass it over to Chris.
Thanks, Gage and Alex. Let's talk a little bit more about understanding the immune system. So one of the major efforts of your lab, and you talked about this earlier, is using CRISPR screens to better understand how our immune system and individual immune cells function. So let's start with your work on understanding genetic circuits that control immune function. Can you give us an overview of that space?
Yeah, so this is something I've been interested in since college. This idea that how do cells take genetic information and use that DNA code inside of their nucleus and how does that control their identity, their collective set of functions? And I do think of that as sort of a circuit board that's inside of the cell that in some ways should be interpretable and understandable. And now with gene editing technology is actually rewirable. But in order to really understand that circuit, we'd like to be able to flip every switch. We'd like to be able to disconnect every wire or turn every switch on and then watch systematically what does that do to the behavior of the circuit. And I think that's the sort of fundamental idea of sort of perturbation-based science, that you isolate one variable at a time and then watch its effect on function. That's how you really understand causality. And CRISPR now gives us the ability to do that, just that, and the idea is that now CRISPR is so easy to deploy that it doesn't have to be one CRISPR edit at a time. We can now do this in one experiment. We can take a collection of human cells, and in each cell, we can disrupt a different gene. Right, we can direct CRISPR to disrupt a different gene, and that's by analogy just pulling apart each wire. There's roughly 20,000 different genes in the genome. You can have a collection of human cells, and then we can just watch and see, okay, if we disrupt that gene, what happens to cell function? And there's a collection of functions that we care about that we can measure in different ways, but that's getting better and better, the ways that we've been measuring technologies. So now the perturbation technologies are extremely powerful. We can test every gene in the genome in one experiment, and we can start having more and more sophisticated measurements. And we put those things together. Some of the way that we've talked about this, the way that I've talked about this, is that I think we're heading towards a Rosetta Stone. And basically each cell sort of fills in the gaps for each other, right? That's the idea of the Rosetta Stone. You can compare side by side two different things. Then we compare and we say, oh, look, that tells us exactly how this language works. This gene is feeding into a circuit that controls this function. And we're starting to really understand the language of DNA elements that control different behaviors. And I think that this does 3 things for us. And many groups are now doing this in different contexts, but we have really focused on deploying this directly as much as possible in the real deal human T cells, so that we can understand not just DNA codes in some sort of abstract way in an experimental system in the lab, but understand them really in the human immune cells that are central for human health and disease. It does a few things. It tells us basic lessons about how these cells are wired. It tells us too about how genetic variation in DNA can affect immune cells. And that gives us a lens to interpret genetic variants that are associated with immune dysregulation. All of a sudden, we start to have the tools to understand genetic variants that might increase risk of autoimmune disease or other types of immune dysfunction. 3, I think it points us to new targets for drugs. This is what, the pharmaceutical industry has this incredible amassed power to go after a drug target once it's identified. But the bottleneck is the risk associated with that. If you pick a drug target, is it going to have the expected effect? And I think the idea of having human genetics and human genomics as a sort of gateway towards identifying really drug targets is extremely powerful. And so in addition to being able to measure DNA sequences in human patients, to couple that with actual perturbation technologies in human cells and really understand the function of individual genes in the cells that we care about is helping to provide higher likelihood candidates for drug target development. And then the final element, and where we've put a lot of our attention, is because we're doing this really in human T cells, we're not only finding targets for drugs, for small molecules or biologics, we're finding targets that are the building blocks for gene engineering in cell therapies. And that as we understand the way that different DNA sequences control immune cell function, we can actually start really designing synthetic programs that turn genes on off or turns genes on to create a logic that really shapes how cells will function. And those genetically engineered cells designed outside of the body with CRISPR engineering could then be infused into patients to have really sort of precise logic gated functions that make them more effective agents for precisely treating disease, right? These should not be the sort of carpet bombers of traditional immunosuppression or chemotherapy. This should be sort of, as we get better and better at understanding these DNA sequences, we should be able to really dial in immune cell functions to direct these immune cells to have precise anti-disease functions.
Wow. Talk about a shift from, as you said, carpet bombing to what sounds like a lot of precision immunology. So as we think about what your lab's doing, you're also using CRISPR to identify factors imparting immune cell fate and function. Can you give us an overview of what's going on here?
Yeah, so I mean, maybe it's worth stepping back, even though thinking I was influenced by when I was a graduate student. I did my PhD at MIT and worked at the Whitehead with two really great scientists as my mentors, Rick Young and Rudy Yanish. And Rick is a world expert in understanding transcription factors and transcriptional regulation using genomic technologies to understand how transcription factors shape molecular programs. And Rudolph was thinking about epigenetics and what controls early pluripotency of stem cells and early cell fate decisions. And I found this really exciting niche at the intersection of these two labs, thinking about how do key transcription factors or regulators either push a cell to be pluripotent or start to take a pluripotent cell and start pushing it down one lineage or another. And I still am fundamentally interested in that. Of like, what are the key natural switches or now synthetic switches that determine what state a cell will enter into, and how much can we really shape those decisions that a cell can potentially make. And so I really see much of our effort as extending that now, not into stem cells that grow in a dish, but into human immune cells. Can we take an immune cell and really think about the full set of states or identities that it might have as a pro-inflammatory cell or different types of inflammation versus a regulatory T cell that will dampen down inflammation? What determines which one of those identities a T cell will have? How can we shape what types of inflammation a T cell might drive, or how effective it'll be at treating autoimmune disease and dampening? So, this set of questions of what are the regulators that determine the identity of T cells tells us fundamental things about how these T cells function in health and disease. And now is not just, curiosity-driven research, but is actually will be fundamentally enabling to think about how do we pull those levers inside of cell therapies to tune the fate and function of a genetically engineered cell therapy.
a lot to think about, especially as we start to unravel that complexity that comes with immunology, given the dynamic states of the cells as you just described. So obviously, immunotherapies are an incredibly hot area, and we'd love to understand, and you've touched on this a bit, how will this improved understanding of the immune system and the cells that make it up improve or change how we develop drugs, especially because we're not going to go from 0 to 100, but there are going to be stages along the way.
Yeah. So I think that, well, I think immunotherapy is often talked about in the context of cancer therapy. So let's start there. But I think that this type of thinking is not limited in any way to cancer immunotherapy. But the cancer immunotherapy has been so exciting and fast moving. And I should also say, when I was going through medical training, I finished my residency in 2012, so about 10 years ago. Cancer immunotherapy in the places that I was trained was a fringe science. It was heavily discouraged, and we were told not to go down that road. I think clearly there were the Jim Allisons of the world who were pushing hard on this and have been proven right by time. But this was not the world in which I was trained 10 years ago, and that has dramatically changed. This is now very much clearly mainstream medicine and science, thanks to the unbelievable successes. So I think in cancer immunotherapy, we've really seen two flavors of this. We've seen drug-based immunotherapies, where especially the checkpoint blockade, where you take the brakes off of T cells that are in the body. They're naturally occurring, but they're not able to fully clear disease because of the brakes that have been the negative regulators that are on those T cells that are preventing them from being fully functional. And drugs can unleash their power, right? So that's one area of immunotherapy. I think clearly we'd love to find more checkpoints or checkpoint equivalents where we can find drugs to unleash the power of the immune system and direct it against cancer. The other area is the dramatic success of cell therapies, right? And this is now comes in the form of a number of different FDA-approved CAR T cell therapies. The current ones are the cells come out of the body, they're genetically modified outside the body with viral vectors. They get infused back into a patient through an IV drip. And for certain types of blood cancers, it's really unbelievable how successful this has been for treating some patients that would otherwise not have had treatment options. So I think, I think about immunotherapy in both of those flavors. Can we find new drug targets that will unleash the power of an immune cell, especially right now thinking about T cells, but other cells in the immune system? Or can we find ways to genetically modify those cells? And I don't think we have to be limited to viral vectors anymore. Now we're thinking about Crispr therapies, where we can pick a site of the body, a site of the genome, and rewrite it. So we have the precision, we move away from viral vectors. Some of this can be done outside of the body for very precise CRISPR editing. We and many others are also thinking about, can some of these edits be delivered inside the body to increase access and reduce the manufacturing bottlenecks? So, but I think those are the two flavors that we're thinking about of, can we find drug targets or can we find gene and cell therapy targets? And as I said, that's not limited to cancer. We're thinking a lot about extending this same kind of logic to autoimmune diseases. I've been interested in, since grad school, in regulatory T cells. And regulatory T cells have the opposite function of most T cells in the body. Their job is to prevent excessive inflammation and prevent autoimmunity. And so those cells actually go around and they can dampen down inflammation. They can promote healing in a number of different ways. So can we make better regulatory T cells? Can we direct regulatory T cells to the right side of the body? Could regulatory T cells deliver payloads to treat inflammatory diseases? And we're thinking a lot about what's the genetic engineering that we can do to a regulatory T cell to make them into more effective cell therapies. We're thinking about infectious diseases, I think all of us have been inspired by mRNA vaccines, and really sort of like another arm of what I think of as genomic medicine, of using mRNA genetic programs to instruct cells in the immune system to protect us. Can we extend that to think about other ways that we can engineer genetic programs and use immune cells to protect us from serious infections? And then we and others are sort of starting to think about, like, well, maybe there's some things that the immune system isn't really so good at treating currently, but we don't have to be limited to just the natural function of the immune system. All of a sudden, the immune cells might become like the drug delivery system of the future that can be where we can program in logic. And like once we can really think of, I talked about like understanding the DNA code to understand what makes these cells behave. Well, maybe we can think about making cells that we just program logic into. Of, okay, we want them to go to this part of the body, be able to sense whether there's something happening there, and if they sense something, they release something, or so I think we can start programming behavior into cells to make them kind of little logic robots, biological robots that survey the body and help maintain health. So, that's the most science fiction version. So I think there's a spectrum of what I talked about, everything from improving current CAR T cells and improving access, which I expect is already happening, and we're going to see a lot of that in the next couple of years, to over the coming decade, sort of imagining what the full range of genetically engineered immune cells might be doing to improve human health.
And I think that segues us perfectly into our next topic on T-cell therapies. So I'm going to pass it back over to Cage, because I, for one, would love to hear more about this science fiction.
Yeah, awesome. Thanks, Chris. So kind of you started touching on this, but in addition to kind of your research to better understand the immune system and identify potential targets, your lab is also working with immune cells to develop new therapeutics. So to start us off, can you tell us a little bit more about your work and what you're working on with T cells?
Yeah, we're doing a few things, but I'll give you some snapshots of some things that I hope will enter into the clinic in the next year or two, things that we're working on academically. So there is we're doing so that, one approach where CRISPR can be extremely powerful is in rare genetic disease. And, we're not going after it because it's rare. We're going after it because in certain rare genetic diseases, we know exactly what needs to be fixed with CRISPR. We know which part of that DNA code would benefit if we can fix it. So we've been thinking, where are there opportunities to connect that to this kind of cell-based therapy? And a number of years ago, a colleague at Yale, Kevin Harold, reached out to us and asked if there was an opportunity to collaborate to see if we could help a family that has what appears to be a monogenic form of immune dysregulation. They have 5 kids in this family. 3 of them seem to be affected by different types of immune dysregulation. And genetic sequencing, exome sequencing, revealed what the mutations are. They are ultra rare mutations in a gene called IL-2RA. It's also known as CD25. It's the high affinity receptor for IL-2. And it's found, gene is used in a few different cell types, but it's found at very high levels in regulatory T cells. And so it turns out that the children in this family who have inherited 2 mutated copies of IL-2RA have cells that kind of look like regulatory T cells, but they're not working properly because of this mutation. And UCSF is arguably the world leader in regulatory T cell therapies, just thanks to Jeff Bluestone's work over many years. There's a manufacturing facility at UCSF that knows how to grow up regulatory T cells and deliver them to patients. So we wondered whether there was a chance to bring CRISPR together with regulatory T cells to fix the regulatory T cells in this in the children in this family. And sure enough, we've been getting blood samples mailed to us from these children, and we can actually go and fix one of these mutations and restore meaningful levels of IL-2RA to the surface of the regulatory T cells. And so one of the things we're very actively working on, this is a partnership for many years with Jennifer Doudna, started an institute called the Innovative Genomics Institute, where I'm the scientific director of human health. UCSF also more recently started a living therapeutics initiative to try to advance cell-based therapy, cell and gene therapies. And so we are working across all these groups to see if we can actually create a gene-edited regulatory T cell. This will be the first to my knowledge, the first CRISPR-corrected regulatory T cell therapy. And we're going to try to do this in N of 1. I hope it is actually a way of treating the immune dysregulation in the oldest affected sibling in this family. But I also hope more broadly, this gets our foot in the door to start thinking about how can we use CRISPR to start engineering regulatory T cells to treat autoimmunity, inflammation, help patients who are undergoing organ transplantation, so we can get targeted tolerance rather than broad immunosuppression. So that is a major endeavor. We're also just thinking with the Living Therapeutics Initiative and with the Innovative Genomics Institute and with a new institute that I launched in the past year or two called the Gladstone-UCSF Institute of Genomic Immunology. Can we broadly start thinking about using CRISPR-based cells to treat cancer? And we're going to start with CAR T cells, but we're moving rapidly to think about, what are all the ways that CRISPR can be used to enhance T cell therapies for cancer? So we're thinking broadly about different classes of disease. How can we be innovative in our manufacturing capabilities? And then, Chris, to go back to your question, how do we take everything that we're learning from the high throughput CRISPR screens and have a pipeline to start introducing those into version 2, version 3 of these cell therapies that we're delivering, so that we continually are iterating and making better and more powerful cell therapies. So you need to have both the discovery pipeline and the manufacturing pipeline. When we put these together into this interdisciplinary academic playground that we're creating, I think we're going to move more and more rapidly towards whole new ways of treating human disease.
That's awesome. I think those are some great missions. I really hope it works out. So you've talked a lot about kind of using CRISPR to start creating these T cells for immunotherapies, particularly in oncology. How do you think that using CRISPR and the approach that your lab is taking can improve upon the things that are already out there?
Yeah. So I think there's a lot to say here. I think that we've really been inspired by the power of existing CAR T cells. And the way that current CAR T cells are made is, as I mentioned, with viral vectors, retroviruses or lentiviruses that are really effective. These are sort of engineered viruses that take advantage of the DNA delivery capability of viral vectors to introduce genes. But there's a few problems. They introduce genes into non-targeted sites in the genome, so you can't really control where that extra DNA is going in. And also, viral vectors they take a long time to make, and they're really expensive to make. So we have put a lot of effort into using CRISPR to circumvent both those things, to get targeted DNA introduction, to pick a site in the genome and rewrite it with CRISPR, and then also to move away from the need for viral vectors so that we can introduce new products more rapidly and hopefully more cheaply. I think that hopefully that manufacturing gives us a big leg up to make more powerful products. I think there's this, something that's been pointed out to me that I've been talking a lot about is that 2012, which, as I said, happened to be the year that I finished residency and was moving to UCSF and starting to think about what my lab would study. 2012, two things happened that were really converged and continue to converge in an amazing way. Emily Whitehead was the first pediatric patient who was treated by CAR T cells. And she was going to be sent home on hospice, but instead got an experimental CAR T cell treatment at Penn, and this trial led by Carl June, and she's been cancer-free since then. So that really showed us the power of cell therapies. And 2012 was the year that Jennifer Doudna and Emmanuel Charpentier published a paper in Science that showed that CRISPR was a technology that could be used to edit DNA sequences in a flexible way. And so I am just continuing, this is just one of these magic things of two great things that play well together, of the ability to now use CRISPR technologies to make better and better cell therapies, and that continues to be a space that I think just has an enormous, although many people have now joined us in these efforts to bring CRISPR together with T-cell therapies, I still think we haven't reached the full potential of everything that is there, of using CRISPR to understand the DNA programs, using CRISPR to manufacture more precisely these cell therapies, and then putting these things together to start introducing synthetic biology with CRISPR into targeted places in cell therapies. So, I think that nexus continues to be an incredibly rich place for innovation and exploration.
Yeah, I totally agree. And actually would love to dive in a little bit and talk a little bit more about what you think, the full potential for T cells look like. You mentioned some of the scientific applications, and some of those seem like they're coming to fruition. A couple of days ago, researchers published a paper about CAR Ts used to reduce fibrosis and heart failure. Something which is incredibly cool and, not what you would have thought of as a traditional application for CAR T cells. So I'd love to hear, where do you see these T cell therapies going kind of in the next 5 years? And then more broadly, where do you see immunotherapy, specifically cell-based immunotherapies, like kind of down the line in the future?
Yeah, I'm glad you brought up this paper. It's a really exciting piece of work. And it's exciting, well, for a number of at least two different reasons. So this was from the Epstein and June labs and others at Penn, and they had previously showed this thing that I think got many people excited, which was that CAR T cells could be directed against fibrosis in heart failure. Now, what this latest paper shows is not only that there's continues to be promise in that area, but that they in addition to that, expanding the reach. So the first area is expanding the reach of CAR T cells beyond cancer into diseases that are not, where you might not even want to have broad T cell killing function, but just a little bit of targeting of these T cells could potentially restore the human health. So that's one, is moving beyond the CAR T cells, moving beyond cancer. The second complementary reason that this is so exciting is that it's a delivery paper. And this paper uses the power of nanoparticles to deliver a temporary instruction to T cells inside the body to convert them from a normal T cell into a CAR T cell, but just for a little while. And I think that the fibrotic application is a really cool application where a little bit of CAR T cells may go a long way. And so this sort of transient in vivo delivery really opens up a possibility of thinking about how will we treat patients in the future and for different types of disease. And of course, this beauty of doing this inside of the body means the patient doesn't have to go give their cells through an extended blood draw. The cells don't have to be genetically engineered in a factory in a kind of artisanal, patient-specific way and then reinfused. You're really just injecting the nanoparticles. And I now have had 3 rounds of nanoparticles injected into my arm to deliver mRNA to protect me from COVID, which is great. But now a similar next engineered version of that could actually be used, not for gene engineering, no permanent gene engineering, but just to give this transient instruction for a cell to take on a new temporary function of becoming a CAR T cell to treat human disease. In the case of this CAR T cell for fibrotic disease. So I think we're thinking a lot about both of these aspects of what else can CAR T cells be made to do? Where can we push them to treat different diseases? And what are the new delivery modes? And this is something that we're doing a lot in collaboration with Jennifer Doudna and the Innovative Genomics Institute of what are going to be the innovative ways of delivering CRISPR to cells inside or outside of the body in a targeted way. I've also been involved in setting up a company called Spotlight Therapeutics that is a company dedicated to in vivo delivery of CRISPR molecules to really make CRISPR into a biologic medicine. So not something that has, that has to be done in a complex manufacturing process, but that would be an injectable biological agent, not for germline editing, but for targeted somatic cell editing to make CRISPR into a drug.
Oh, that's amazing. I think time for one more question on this topic before I move on. I'm kind of just curious to hear, are there any really interesting applications of CAR T or kind of delivery technologies or other emerging technologies in the CAR T space that you think are particularly interesting right now?
Well, I think we've touched on a few of them. I mean, I think this idea of, broadly using CAR T cell CARs, or maybe it's going to be other types of receptors as ways of addressing cells to a particular type of the body, right? And so, like we talked about this now extending into fibrotic disease. What else can we really find ways to decide where a T cell will go? And I think if we can do that, then we can start. All of a sudden, T cells become something that can go where we want them to go. And so I think many people are thinking about like this kind of cellular addressing for immune cells is one area. I don't have an answer yet, but I think people will think, can we send cells into the brain? Can we send cells into different tissues? And then for each disease, for each organ, there are disease opportunities of what we can make those cells do once they get there. The other is like, are CARs just limited to T cells? And I think that another really interesting area is like, how far out can we branch? I talked about the idea of, well, maybe we can start genetically engineering regulatory T cells, so not just the conventional cells, but regulatory T cells. But beyond that, I think there's Sar-Gill and others have thought about, can you start engineering receptors into myeloid cells, it's cells that aren't the T cells, but are other parts of the immune system. And I think, thinking outside the box of, what other cells can we start putting synthetic programs into to transform them into cell therapies? We're still in the early days of imagining the full set of possibilities of what we can make cells do to have new properties and have new ability to transform them into drugs. I mean, right, this is like, I would say, when I say that a little quickly, this idea of transforming a cell into a drug, I think that this is like, all of a sudden there's a new way of thinking about like these things that I learned in med school, like pharmacokinetics, pharmacodynamics, like, what are the drug properties of a cell? What's its half-life in the body? What's its biodistribution? And all these things might be things that we can really control either by changing the initial cell, right? Some cells may be more long lasting, others may be short lasting. So you can have like the initial cell that becomes like a menu of different starting points, but then we really will have like an enormous list of how we engineer these cells to give them specific pharmacological properties to treat different diseases.
I totally agree. I think the potential of T cells for therapeutics, even moving beyond that, getting cells to start doing things in the body that we haven't even really thought of yet, is going to be a fantastic area of research over the next couple of years. And I'm really excited to see what your lab and others come up with. But for now, I think I'll pass it over to Chris to finish off on our last topic.
Great.
Thanks, Gage. To go back to something you mentioned at the very beginning, Alex, around the infectious disease space. Let's talk a little bit about host pathogen interfaces. So as your lab probes the genetics of the host pathogen interface, can you give us sort of an overview of what you're working on?
Yeah, I'll tell you where we're thinking about this the farthest along. And this is a sort of a special opportunity for me, having trained as an infectious disease doctor, to think really sort of connecting my interest in immunology and gene engineering back to diseases where I spent years seeing patients. For a number of years, I had an HIV clinic that I ran. And so we're thinking a lot about this in the context of HIV. HIV is a virus. There's still almost 40 million people around the world infected with HIV. It's primarily targeting T cells. And so we have really been interested in, can we use CRISPR to understand how this virus with only a handful of genes co-opts T cells and infects them, integrates into the genome of those T cells. How does it hide out latently in T cells to avoid existing drugs and then spread to other cells? So all of that really depends on the interaction of the HIV machinery with the machinery inside the human T cell. And now, we've been inspired, for example, by work of a very close collaborator, Nevin Krogan, who's at UCSF and the Gladstone with us, who has spent a long time studying protein-protein interactions between viral proteins and human proteins. But CRISPR now provides an opportunity to complement these protein-protein interaction maps that were made by biochemistry and mass spec with functional maps, with genetic maps. So now we can take that as a guidepost and start deleting the human gene and seeing exactly the rules again in these perturbation studies of which human proteins is HIV actually depending on, and which ones actually in some cases restrict HIV. So there's a dance between HIV and the T cells. So I think that this is going to tell us again, like lots of lessons about how does a pathogen interact with a human cell. But this is an instance where I really am passionate about the possibility of finding new drug targets. And I just want to quickly say my perspective on where we are in HIV. There's a number of efforts to do cell therapy for HIV. I hope they work. People are trying different things, including CAR T cells for HIV, trying to make immune cells that are resistant to HIV infection. I hope they work. I think, though, that that's not what we need to really turn the tide of where we are in this epidemic. We already are at a place where we have pretty good drugs that in many cases can be given once a day. But HIV remains a chronic disease, and you have to take those drugs for the rest of your life, for a patient's life or an individual's life, an HIV positive individual's life. We need a way to get rid of HIV as a chronic disease. And there's really only two ways to do that, and do that at the scale that can be really address this worldwide epidemic, including in places where resources are limited. We either need a vaccine to protect people from getting HIV, or we need a one-time cure. And we need a one-time cure that would be affordable and accessible to people around the world. So what's my hope here and my aspiration? I hope that CRISPR can find the drug target to tell us what's missing in our current HIV cocktail. And so we've really been focusing in on this. We got a gift from Gilead, but we're continuing to push to really use CRISPR as a tool to exactly understand which parts of the T cell machinery keep HIV in a latent state. And if we can really understand that, if we can use CRISPR to precisely understand what controls latency, then we have a hope to design new drugs, either to keep HIV permanently latent or to coax it out of latency and so that it can be susceptible to other existing antiretrovirals. So early, but aspirational, that we are heavily interested in seeing if we can use CRISPR as a tool to find new effective ways to cure chronic HIV and perhaps other chronic viral infections.
And I would honestly be curious to see how that latency could be translated, as you say, to other chronic or even acute potentially infections. That could be that's not something I've thought about before. So thank you for bringing it up, and for all the really great work you're doing. Just in the interests of time, thinking about things in the light of the current pandemic and potentially future pandemics, and I guess disease more broadly, how do you choose problems to try and tackle? What is the sort of broader application of this work?
It's a great question. I think the approach that I don't think there's like a right answer to this. I think it's great that scientists take different approaches where some people go deep into one disease and really sort of know everything about it. That hasn't been our approach. Our approach has been to try to understand the immune cells and T cells in particular. And I think that that has been in many ways like surprisingly, even going into it, it's been surprising how rich that's been, to see that T cells on the one hand are the drivers of autoimmunity and excessive inflammation. If we can understand what controls that, we might find new targets for multiple sclerosis and Crohn's disease and rheumatoid arthritis. So I really wanted to understand the genetics of what shapes the risk of autoimmune disease. It was sort of a surprise to me then to think, okay, well, as we're developing the tools to understand that genetics, maybe we can also start thinking about engineering T cells to be better at treating cancer. And so the unexpected crosstalk between interests that started off and towards autoimmunity and how that, focusing in on T cell genetics might also be useful to drive cancer immunotherapies. And then circling back and thinking about host pathogen interactions in T cells and other immune cells. So, we continue to really think about how well we can understand the T cells and what are the perturbations, either with drugs or with genetic engineering, that we can use to push T cells in different directions to treat different diseases. So the cells become the hub of how we think about our research and then look for applications around that.
That's great. And Cage, passing it over to you just for a few closing questions.
Yes, thank you so much, Alex. This has been a fascinating and enlightening conversation. But before we go, a few last questions to round things off. First off, where can people learn more about the work you and your lab are doing?
We have a website, the Marson Lab website. We also, I launched about 2 years ago a partnership between Gladstone, which is a small independent research organization, and UCSF, which is a major state academic medical facility. This Gladstone-UCSF Institute for Genomic Immunology. And the idea is really to create a collection of labs that are working across disciplines, integrating genomic technologies, genome editing, functional genetics, computational biology, all the way to cell therapy manufacturing, clinical delivery. And so if people are interested, continue to look out for our information about the Gladstone-UCSF Institute of Genomic Immunology. Reach out to us if you're interested in collaborating or supporting us or partnering in various ways. The possibilities are enormous, and we're really eager to work with people who are excited about this area of science and biomedicine.
Fantastic. And last thing before we go, do you have any shameless plugs or closing thoughts for us?
I would heavily, plug for this institute. I think I guess I'll supplement that with plugs for a number of different companies that I'm involved with. I helped to co-found Spotlight Therapeutics, which I mentioned. I co-founded Arsenal Biosciences, which is doing CRISPR programming of T cells to treat cancer. I'm developing a new company called Genomics to advance new spatial genomic technologies. So I'm heavily involved with those and other companies. But even with that, there's something that is totally distinct and special about the academic sphere, where we have the opportunity to explore freely, to go back to the beginning of this conversation, to really push the boundaries of our understanding as humans, and to think about the outer edges of where that might lead us to new opportunities to treat people, to help people. And that truly depends on philanthropic support, government support and funding, but for it to really have the full ability to translate that into new therapies requires visionary philanthropic partners. And we're looking for those people all the time. So people who want to be part of something new and exciting that has a huge potential to help people, we'd love to talk.
Amazing. Well, thank you again, Alex, for an absolutely incredible episode. This was a lot of fun, and we're incredibly grateful for your time.
Thanks for a great conversation, and thanks for having me.